Are we good?
Oh, yes.
Okay. My name is Patricia Cockett. I'm a PhD candidate at Texas A&M University in Corpus Christi. I'm also a scholar with the Center for Coastal and Marine Ecosystems. Make sure you tell Dr. Kelly I nailed it. I'm going to share some of my research with you guys today. It's kind of boring. It's not nearly as entertaining as you guys just saw, but I think it's important. So what I did is I compared biological responses to long-term climate trends in our 3 South Texas estuaries. So there's a need to link our climate, freshwater inflow, and biological resources in South Texas estuaries. They maintain a lower salinity that's crucial for the productivity of the estuary, delivers nutrients and sediments to the estuary, and it provides the necessary salinity balance that supports the nursing grounds for marine life and economically important fishery species. So I like to eat fish, and I need fresh water for their nurseries, so it's really, really important. So right now, the conceptual model for managing our freshwater inflow, it's this domino theory, right? And so, you know, you start out with your freshwater inflow, with the timing, frequency, and duration of your river discharge. And then that causes a response in your estuarine conditions, right? So you'll see changes in salinity, you'll see more sediment, dissolved material. And then you'll see a response in our estuarine resources, right? This is where we get all our fish from. But they forgot one thing, which is climate. So we're gonna throw that in there. Also, my advisor is Paul Montagna, and he's like a superstar when it comes to he's like a freshwater inflow guy. He's like a salinity guy. And he really pushes this estuarine conditions, because people used to skip from freshwater inflow to resources all the time. So it's so important that we look at this one. So in Texas, we have a climatic gradient along the Texas coast. So it's much more, there's a lot less saline. The salinity is much lower in the northeastern region. And as you go further south and west, it'll start getting more and more salty. And our school is right here. That's where our university is. And so I'm gonna be looking at 3 of our bay areas. So the Nueces Bay, Nueces Estuary, Guadalupe, and Lavaca Estuary. Pollock et al. In 2011 had found a relationship between the Oceanic Nino Index and the North Atlantic Oscillation. And she found that correlation with the abundance, biomass, and diversity of benthic in fauna. So all the little bugs that live in the mud. So what happens is the sea surface temperature gets really high in the Pacific Ocean. And then, you know, the water, it goes up, our moisture goes up, and it travels over jet streams, and then it rains on Texas. Now, the same thing kind of happens with the North Atlantic Oscillation, but we don't quite know exactly how it's affecting it. So as salinity decreases, we see an increase in abundance, biomass, and diversity with our benthic and fauna. And one of the alumni, Megan Hardigree from my lab, she compared all 3 of the bays that I also compared. And she found an increase in temperature over time and a decrease in dissolved oxygen. Okay, follow along with me in this graph. I stole this from her draft of her master's thesis. She graduated in 2017. So it's a little small, but we can work with it. This top row is the Lavaca, Colorado estuary, and that's the most northeastern one. So you expect, lower salinity. Guadalupe is kind of in the middle. And the Nueces estuary is on the bottom over here. And so what we're looking at is the diversity, biomass, and abundance of benthic and fauna. And we see a decrease. Oh, this top line, this is our secondary bay. So it's the one that's closest to the river mouth. And then on this bottom one, this is our primary bay. And that's the one that kind of feeds out to the ocean. So we see decreases in diversity, biomass, and abundance for all three of these in the Lavaca estuary. In the Guadalupe estuary, you don't see much of a trend. And then it starts getting weird, where you see a decrease in one bay and an increase in the other bay. And for our abundance, you just see an overall decrease. In the Nueces estuary, you see an increase, right? In all three of these variables. Maybe not this one. And so it is a different system. So now another alumni, Elaine Kirk, she studied, what she did was she broke up each one of these bays and she looked at epifauna. And what she found was that in times of drought, you would see an increase in the abundance of epifauna in these upper reaches of the estuary. So I decided to focus on these upper reaches. So this section being section NC1, NC2, so the ones and twos. So here's our new model. And the purpose of this is to compare like complex climate, hydrological, and biological interactions among all these bays. And so I threw in climate, there's our oceanic Nino index and our North Atlantic oscillation. And for our freshwater inflow, I wanted to use of the variation over time of different freshwater inflow events. And then we have our species abundance, and I chose blue crab and white shrimp. All right, so Sabo et al., he quantified all these cool metrics, and he was able to predict the right amount of fresh water for a fishery in, I think, the Mekong Basin. So what I did was I took what he did and I adapted it to like monthly. And he also used this multivariate autoregressive state space model. And it's a linear model. So this is kind of like your regular linear regression, right? Y equals mx plus b. So data enters the model as y, and then it feeds into this autoregressive part. And so b is our growth rate, and we can actually throw in covariates. It's not a predictor because time is our predictor, right? This is for time series. So our covariates will be our indexes and my hydrometrics that I quantified. So it's multivariate because I actually have to build all these matrices, and this is a really small example, and you don't have to worry about it or think about it. The only thing that's hard is like actually typing in, making the matrices. That's the wish. All right, so I had to break it up. So I ran my model for the first part of my entire model. So here I have all my hydrometrics, right? It enters as y. Then I have my covariates. Oh yeah, that's right. All right, so what we can do is we can actually test hypotheses to see if different time series, share the same variance. And you can do that with your little Z model. But then you can test the effects of your covariate on your variance. And to do that, all you gotta do is take Z out. So it's super cool. So the data I used was the climate indices, monthly, daily discharge from USGS. And I got 2 rivers from Nueces, one from San Antonio, one from Guadalupe. So these 2 right here feed into the Guadalupe estuary, right? 2 river basins. And then I have the Lavaca River that feeds into the Lavaca one. Now this Nueces River up here, I have 2 of them because I wanted to look at it. I figured I'd show it to you guys too. So we only have data from 1990 to 2016, but our biological data starts in 1992. And Cal Allen is much closer to the bay. So it's more like what you would see entering the bay. Talking too much, I gotta go. All right, so I calculated a seasonal baseline with a discrete fast Fourier. This is an example of 1990. Quantified some stuff. So here's our high monthly anomaly magnitude. Here's our H staff. I wish I could tell you guys what the this is pretty obvious, like slope for each month. We didn't use that one anyway, or this one. Okay, then I did correlations between our climate indices to pick the lags for each one. I went for a model, right? Maximum values. Then I called for collinearity. So here are results. So this is my hypothesis, right? This is what won after the model. They're all different. If they were animals, you'd say subpopulations. And then our second place, these two shared the most, and these three shared the most, which is kind of what I expected. For H staff, we saw grouping in Guadalupe and La Vaca, and the rest were different. And second place again was our individual variances. And then the covariate effects. So these are the effects of NAO on our hydrometrics and ONI. And so we see there's some significance there. It's increasing. It has a positive effect on this. Here's another one. First place, same thing. We're seeing the same trends again. Yeah, they're kind of the same. We'll probably correlate a lot. LSAM. So this is a little bit more of like a low flow kind of thing, right? So red or low flow. Same trend here, same trend again. As far as effects go, all it does is it brings up that low magnitude, so you don't see such drought conditions, as we call it. For our multi-low flow intervals, same trend again, it's looking pretty good, but it has a negative effect on these things. All right, next model. So I remember I said it was like really important to include this. I kind of just skipped over it because I couldn't find the right data. But my boss said it's really important, so you should remember that. Right, so here we go, comparing blue crab and white shrimp. And they all have subpopulations, right, separate populations. But our second runner-up pairs them by their upper estuaries. And then a third one paired these two and thought Nueces was different. And then here are effects of our monthly metrics on our population. So we don't see a whole lot of response coming from the blue crab. But if you look at the white shrimp, you see more of a response, right? So we see this blue dot, that's the upper reaches of the estuary. It's kind of cool, right? The red one is the lower part of the bay. And so we can see that these have like positive or negative effects on their population. Conclusion, hydrologic metrics among river basins, they're all different. ONI and NAO, they all affect the hydrologic metrics different. Yeah, they affect the different river basins differently. The frequency is affecting river basins differently. Blue crab and white shrimp within and among secondary and primary bays respond differently to factors different by climate. And then our ecosystem responses are essential to understand how fisheries respond to hydrological vectors. As we saw, blue crab, we couldn't really see very much at all. So we kind of need that in between part. Implications, we should look at this separately again. Future endeavors, I want to take out noise, and I'll probably add that ecosystem part because it's really important. Thank you, everybody.
We have time for one quick question.
Hey, Colin, good to see you. I'm glad you will.
I know, thank you.
Yeah, it's been so long. I wanted to ask you, what challenges did you experience? It seems like you use a lot of big data applications and databases. What challenges did you experience if you were looking to choose the particular model that you use and the organization application?
The hardest part is searching and setting initial conditions. And then like bootstrapping and kind of making it efficient so they can run it. Otherwise, it'll take like a week. And it's always big.
Thank you.